Have you ever wondered what goes on inside the tiniest parts of your body? Deep within our cells, there is a control center called the nucleus. This is where our genes live. Genes are like instruction manuals that tell our cells what to do. For a long time, scientists have been trying to solve a big puzzle. They wanted to know how special proteins turn these genes on and off. It's a bit like flipping a light switch. Many of these important proteins are surprisingly floppy and don't have a fixed shape. Imagine a spaghetti noodle instead of a solid brick. It was a real mystery how these wiggly, unstructured proteins could do such a precise and important job inside our busy cells. These special proteins are called intrinsically disordered proteins, or IDPs. The name sounds complicated, but it just means they are flexible and don't hold one single shape. For many years, scientists believed that proteins needed a very specific, rigid structure to work correctly, like a key fitting into a lock. This idea was a cornerstone of biology. So the discovery that so many proteins involved in controlling our genes were actually disordered was mind-boggling. It challenged everything we thought we knew. How could something so seemingly messy and chaotic be responsible for the incredibly organized process of switching genes on or off? It was like trying to build a complex machine using only pieces of cooked spaghetti. Scientists had some theories, of course. One popular idea was that these floppy proteins would briefly fold into a more solid shape right when they needed to interact with DNA or other proteins. Perhaps they were only disordered when they were floating around by themselves. Another idea was that their very flexibility was their superpower, allowing them to grab onto many different partners and perform a variety of tasks. But these were just ideas. No one had a clear picture of what was really happening inside the cell's nucleus to bring all these floppy, important proteins together to get their work done. The real mechanism remained hidden from view, waiting for a new discovery to shine a light on it. This puzzle is super important because controlling which genes are active is fundamental to life. It's how a skin cell knows to be a skin cell and not a brain cell, even though they both have the same set of genes. When this control system goes wrong, it can lead to serious problems, including many types of diseases. So, understanding the basic rules of how these floppy proteins operate wasn't just an academic question, it was a critical piece of information that could unlock new ways to understand and potentially treat illnesses. The scientific community was on the edge of its seat, searching for the missing link that would explain this hidden order within the apparent chaos of the cell. Then, a breakthrough happened. Scientists made a fascinating discovery that changed the whole story. They found that these floppy, disordered proteins don't just magically organize themselves. They need help from a partner. This partner is a structured, well-organized protein called beta-catenin. Think of beta-catenin as a solid, reliable docking station. It has a specific grooved shape that acts like a landing strip. The floppy proteins, which are normally just wiggling around, can come and attach themselves to these specific grooves on beta-catenin. This was a huge aha moment. The chaos wasn't chaos at all. There was a hidden organizer bringing everyone together to get the job done. This discovery was made using some really cool and advanced scientific tools. Researchers were able to look at these molecules in incredible detail, almost like having super-powered goggles that could see things at the atomic level. They observed that beta-catenin doesn't just bind to one floppy protein. Its surface is large enough to act like a scaffold, a kind of molecular construction platform. It can hold multiple different floppy proteins at the same time. By docking onto beta-catenin, these disordered proteins are brought close to each other. Suddenly, they are no longer floating around randomly. They are gathered in one place, ready to work together as a team to switch on a gene. This idea of a structured scaffold organizing disordered partners is a completely new way of thinking. Before, we imagined proteins interacting one-on-one like two people shaking hands, but this is more like a school bus driver. Beta-catenin picking up a bunch of different kids, the floppy proteins, and bringing them all to the same school, the gene they need to control. Beta-catenin doesn't do the main job of gene switching itself. Instead, its crucial role is to be the organizer. It provides the structure that the floppy proteins lack, allowing them to function as a powerful and coordinated group. It's an elegant solution that nature came up with. So, the mystery of the floppy proteins was finally starting to make sense. Their flexibility allows them to bind to the grooves on beta-catenin, and beta-catenin's rigid structure provides the stability and organization needed for the next step. This process creates what scientists call a transcriptional hub, which is a fancy term for a temporary command center built right next to a gene. It's a beautiful example of teamwork at the molecular level. A mix of structured and unstructured parts come together to create a functional machine that is much more powerful than any of its individual components. This discovery has opened up a whole new field of research. This discovery isn't just an interesting science fact, it has real-world importance, especially in cancer research. Let's look at one specific example: adrenocortical carcinoma. This is a rare but very aggressive cancer that affects the adrenal glands, which are small glands that sit on top of your kidneys. Scientists have known for a while that many patients with this cancer have a mutation in their beta-catenin protein, but they didn't fully understand why this specific mutation was so bad, and how it was driving the cancer's growth. The new discovery about beta-catenin acting as a docking station provided the missing piece of the puzzle. It gave researchers a whole new way to look at the problem. In healthy cells, the beta-catenin docking station works perfectly. It gathers the right floppy proteins at the right time to turn on the genes needed for normal cell function. But in this type of cancer, the mutation changes the shape of the beta-catenin protein. It's like breaking some of the docking ports on our landing strip. This change means that certain floppy proteins can no longer attach properly. Specifically, a group of proteins that normally act as off switches for cell growth are unable to dock. Without these off switches, the cell's growth signals get stuck in the on position, which is a classic recipe for cancer. The cells just keep dividing and growing out of control. This new understanding is a game changer for researchers. Instead of just knowing that a mutated beta-catenin is bad, they now know why it's bad on a molecular level. They can see exactly which floppy protein partners are being kicked off the docking station. This detailed picture is incredibly valuable. It points directly to the problem. The interaction between beta-catenin and its specific protein partners is broken. It's like a mechanic figuring out that a car won't start not just because the engine is broken, but because a specific wire connecting the battery to the starter has come loose. Knowing the precise problem is the first step toward finding a way to fix it. This deeper insight offers hope for developing new and better medicines. In the past, trying to target a protein like beta-catenin was very difficult. But now, instead of trying to fix the whole broken protein, scientists can think about creating a drug that helps reconnect the floppy proteins to the mutated docking station. Or perhaps they could design a medicine that mimics the action of the missing off-switch proteins. This approach is much more specific and could lead to treatments that are more effective and have fewer side effects. It all starts with this fundamental discovery of how these proteins are supposed to work together in the first place. The importance of this discovery goes way beyond just one type of cancer. The principle of a structured protein organizing a team of floppy ones is likely a common strategy that our cells use for many different jobs. We are just beginning to see how widespread this process might be. Scientists now suspect that similar docking station mechanisms are at play in controlling all sorts of cellular processes, from our immune response to the development of our brains. This single discovery has opened up countless new questions for researchers to explore. It's like finding a new key that could unlock many different doors in the vast and complex world of cell biology. This new model completely changes how we think about developing medicines for the future. Many diseases are caused by proteins that don't work correctly. For a long time, drug discovery has focused on targeting well-structured proteins with clearly defined pockets where a drug molecule can fit, like that key in a lock idea. Floppy, disordered proteins were considered undruggable because they didn't have a stable shape to target. But now, we have a new strategy. We can design drugs that don't target the floppy protein itself, but instead target the interaction between the floppy protein and its structured docking station, like beta-catenin. This is a whole new playbook for pharmaceutical science. Ultimately, this breakthrough reveals a beautiful layer of hidden organization within our cells. What once looked like a chaotic jumble of floppy proteins is now understood to be a highly regulated and elegant system. It shows us that nature uses a clever combination of both structure and flexibility to build complex biological machines. It's a powerful lesson that order can emerge from what seems like disorder, as long as there is a scaffold to bring everything together. This principle might even apply to other complex systems beyond biology. It's a fundamental shift in our perspective on how life builds itself from the ground up. In conclusion, the discovery that the structured protein beta-catenin acts as a docking station for floppy, disordered proteins is a landmark moment in science. It solves a long-standing mystery about gene regulation, provides crucial insights into diseases like cancer, and opens up exciting new avenues for drug development. It reminds us that there is always more to learn about the amazing and intricate world inside our own cells. This isn't just the end of one story, it's the exciting beginning of a whole new chapter in our quest to understand the secrets of life. And that is what makes science so wonderful.